today. I have Dr. Sandeep Raha with me, who is one of our Cytocentric Visionaries. And today we are focusing on his important work. He has a recent paper out about the importance of oxygen for their trophoblast cell system. And we thought it was really important to talk to him and to find out what the most important aspects of this work are, and what other researchers can get in terms of tips and tricks and advice and wisdom from somebody that's experienced in this area. So without further ado, let me thank you again for joining me and ask you, Dr. Raha, what led you to do this work?
Yeah, thank you, Dr. Han. It's a pleasure to be speaking with you. And as you asked, the journey to doing some of this work with trophoblast cells, which are sort of the stem cells of the placenta, was sort of an interesting one. My background is really in mitochondrial biology and mitochondrial physiology. And as we all know, mitochondria metabolize oxygen and are critically dependent on oxygen for their function. In studying mitochondria, I think in vivo, one of the challenges is in the process of harvesting this tissue, you obviously expose it to 21% oxygen, which is different from what the body experiences and the tissue experiences. And so in order to look at some of the mechanisms, a lot of researchers have been moving to in vitro models. Now, in vitro models, unfortunately, suffer from the same fate, is that we work with them under 21% oxygen, which is not necessarily what they experience in vivo. And so what really led us to doing some of this work, and the work we were able to do after we purchased the ex vivo unit from Biospherics, was that we were able to really do this modeling under what we call sort of normoxia for tissues. And there's been some controversy about the terminology, as you well know. What people call normoxia, we now tend to call hyperoxia, because the concentrations are a little bit different in vivo, the oxygen concentrations. And so what really moved us to try to explore this area was the ability to look at some of this mitochondrial biology in combination with in vitro modeling. And the first step of that is this particular paper, which is characterization of the trophoblast cells and their responses in the form of a in vitro model, which tends to emulate, or what we're trying to do is emulate the placental barrier, the maternal fetal interface. And we wanted to characterize some of the critical parameters as a consequence of developing this model under what we think is physiologically relevant oxygen concentrations for the placenta. And normally we consider this to be in the range of 3 to 8%. It's difficult to get an exact number, but that's sort of where we put the value.
Interesting. Yeah, I like that you brought up the point that oxygen is sort of always in ranges, in tissues, and we don't exactly have the best measurement on what a particular tissue oxygen level is. It kind of a fallacy to think that we should know one number for what oxygen should be in one place in the body?
Yeah, I think that's a great point. I think the literature certainly struggles with characterizing that threshold, and I think there are a number of buffering systems physiologically to protect us. And so, while we might want to think that oxygen levels are maintained pretty consistently, I think consistency within a range is probably a tolerable range, is probably where our bodies sit. And I think if we try to get uh, a number, that will lead to a lot of challenges. I think our bodies are designed to adapt with the environment, and the realistic situation is the environment changes quite frequently, actually, probably more frequently than we would want to admit to. And so our bodies are designed to buffer that oxygen concentration, the changes in the oxygen concentration to some extent. And the mechanisms involved in that are what I think we need to understand more clearly.
Hmm. And so, what in your most recent work, was there any surprises there for you?
Yeah, so we had a couple of really interesting findings with our recent work, which really was, as I said, an in vitro model bringing together these trophoblast cells, and we couple them with human umbilical vascular endothelial cells, or HUVECs, to emulate the fetal circulation. So the maternal-fetal interface is an interaction between these trophoblasts and these HUVEC cells. In that process, we characterize mostly the the trophoblast cells, and what we found was that as I said, at reduced oxygen concentrations, or 3 to 8%, we found the trophoblast cells grew at a slower rate, which surprised us a little bit. But the oxygen is known to trigger differentiation mechanisms, so that might be a good explanation. But also, I think one of the things that surprised us most was at those physiologically relevant oxygen concentrations, there was greater trophoblast fusion. So at the maternal fetal interface, the stem cells fuse to form what we call a syncytium. And that syncytium acts as a interface between the mother's circulation and the fetal circulation. And that fusion seemed to be potentiated under lower oxygen. But interestingly, at 21% oxygen, people normally characterize beta HCG as the marker that goes along with fusion. So when fusion increases, beta HCG goes up. In our model, at the physiologically relevant oxygen, beta HCG actually went down. And so we struggled with this. But one of the things we've seen in the cancer literature is that oxygen changes sensitivities, drug sensitivities, and cellular signaling. And so it's possible that in our system, the oxygen level made the trophoblasts more responsive to beta HCG, so we didn't need as much. Now, we would have to characterize that obviously more. We didn't characterize that statement within the context of this paper, but that is something we're exploring moving down the line. And the final piece was really the selectivity of the interface. We did find that with lower oxygen concentrations, or physiologically relevant oxygen concentrations, the ability of small molecules to diffuse through the interface was reduced, which sort of suggested to us that the interface might actually be tighter as compared to what it is at 21% oxygen. So taken together, one of the outcome of this paper was really the physiologically relevant oxygen concentration leads to a more selective and certainly a more fused syncytium. And the molecular markers that are coincident with fusion at 21% aren't necessarily performing in the same way at the physiologically relevant oxygen.
Interesting. So having that physioxia, that physiologically relevant oxygen level, really changed everything.
Yeah, there were certainly profound changes in function at that physioxia concentrations.
Hmm. So, do you think we've kind of been looking in the wrong place? Doing everything in room air, we've been kind of getting the wrong answers for some of this stuff?
Well, I think two things with relationship to that. I think that there are differences. I don't. I hesitate to say it's the wrong place, but I think there are cellular signaling pathways that are responsive to oxygen. There are tissues and cells in your body that are designed to respond to those. And I think we have to understand that the performance of certain cell types has to be better contextualized. So some of these systems will need to be looked at in the context of lower oxygen. And some of the work we've done before that, even under sort of 21% oxygen concentrations, have suggested to us that even the matrix that the cells sit in actually influences the oxygen that they see. And so, looking at the model as a whole, the relationship between oxygen and environmental conditions, as well as the matrix the cells sit in terms of in vitro modeling, I think that needs to be something that is worthy of deeper consideration. I do think that oxygen needs to be something, when you're doing, especially when you're doing in vitro modeling, it should prompt more thought in terms of its influence. And one might even say that, when some tissues, when people harvest them, the oxygen perfusion that they experience, I think, is a factor in functional outcomes. And one of the things that really helped with the ex vivo was the construction of the unit allowed us to do those harvesting processes at the appropriate oxygen concentrations.
So you had unbroken oxygen control. So you didn't have oxygen going up and down, between steps.
And that we actually found, as funnily as that might sound, that reduced our standard deviations a lot, right? That kept the data scatter down, and we noticed that pre ex vivo and post ex vivo.
Wow. So you saw an improvement in reproducibility.
Yeah, yeah. And so I think that harvesting is now, important for us, and we are moved to try to standardize those protocols.
And try to get that pericellular oxygen to be consistent.
That's right. Yeah,
Yeah.
yeah.
Interesting. Um, Here's a question in a sort of totally different vein, but do you think that journals should require reporting of actual in vitro oxygen levels to apply to things like reproducibility from lab to lab, from paper to paper?
Yeah, and I think that statement is probably where I've seen it apply the most. Two things. One is the work that we're doing with trophoblast cells, and the other is a lot of work with cancer tumors also. I think is very oxygen dependent. Um, we certainly know that tumors exhibit an oxygen gradient across the diameter of the tumor, and if it's a large tumor, it certainly impacts the performance of cells in the core versus the periphery. And I think I think it would be valuable to have that information. The challenge is the capacity of labs to report those kinds of information. I think certainly people working in the area need to consider the oxygen as an influence. And it might be, the journals and journal editors should be on the lookout for conditions where those parameters need to be considered. And I think journals are getting more sophisticated as the technology evolves. People are asking for that information. I still think we have a bit to go, but I definitely think it's important to consider.
Cool, cool. So, where are you going next? What are your next plans?
Yeah, I mean, we have a powerful unit now with the ex vivo, and we can look at a lot of uh, questions. I think we certainly want to explore the influence of the oxygen on the placenta. As I said before, at my core, I'm a mitochondrial biologist, and so there are lots of questions related to mitochondrial function and oxygen, not only in the placenta but in functioning muscle, and to be able to look at how the oxygen environment, especially one of the unique things about the ex vivo that we've really sort of latched onto is its ability to temporally and dynamically control the oxygen levels. And so this allows us to sort of emulate conditions of, oxygen depletion, look at cellular mechanisms that are involved in buffering that oxygen level. And so I think we're really interested in exploring some of those questions moving forward.
Interesting. Yeah, I could see some real differences in the oxygen dynamics between trophoblast system and a muscle system, which because of course with like you're talking about like a skeletal muscle system
Yeah, yeah.
because skeletal muscle depletes so rapidly, once you if you halfway through the marathon, you've got a very different set of conditions in muscle tissue than you had at the beginning, whereas in a placental system you'd probably see more constant conditions.
Hmm. Yeah, yeah.
So there's some really interesting models you could get into there.
Yeah, absolutely. And the really cool thing for us in both paradigms is how the metabolism adapts to that change, right? Because it has to.
Yeah, yeah, it absolutely has to. And so bringing immunometabolism, of course, has been an exploding field right now. And do you see that sort of bringing up the interest in mitochondria and metabolism and control of cellular programming by metabolism and in all the other fields as well?
Yeah, I mean, we're particularly interested actually in terms of how the oxygen, well, in general, stress during pregnancy influences the programming of the fetal physiology. And one of the areas that we're focusing in on are the signals that are released from the maternal fetal interface as a consequence of maternal stress. And one of the maternal stressors is oxygen. Oxygen can be depleted and through various processes, and I think that changes the programming within the trophoblast to alter to adapt the fetus, and I find that, and that includes the the immunological and the immunomaintenance pieces for the baby as well. And so the complexity of this mechanism, the adaptation mechanism, is for me is very fascinating.
That is fascinating. And 2020 has been quite a stressful year for everybody, including, I'm sure, a lot of pregnant moms out there. So, so stress could play a role definitely in how this tissue functions. With your in vitro model, can you look at things like drug passage, drug absorption, and passaging of chemicals between mother and fetus as well?
Yeah, the model is definitely adapted or being developed for part of that, and I think that's one of the interesting things is as we move to more complex in vitro models like organ on a chip, which is a very interesting in vitro modeling system. You do have to consider things like oxygen, but our model in particular, one of the things we're interested in looking at is the effects of cannabis exposure during pregnancy on the maternal fetal interface. And so cannabis becomes sort of a stressor in our model, and we look at how that changes the function of that maternal fetal interface, or the syncytium. And we do find that things like cannabis affect the functioning of that syncytium, and in cannabis, especially THC specifically, affects the functioning of mitochondria within that maternal fetal interface. And so I think bringing oxygen into the equation as a influencer of the outcome is really important. And we do this in steps, but we will be going in that direction as well. And I think you're right. I think it is really important to consider how things like that affect transport of molecules, release of hormones. That becomes an important consideration.
Wow, this is such important work. And I want to be respectful of your time here, so I don't want to take too much longer. Is there anything else that you would like people to know about your work?
Yeah, I think the really the big thing, the big message would be for people to really think about as they develop these more complex models, and we are moving in that direction, trying to reduce animal use, trying to develop more human relevant models, is that we do need to consider the matrix as an influence on cellular function as well as oxygen, and sometimes those are both related. So I think that would be what I would like to sort of leave you with, is that for people to think about those two influencers in looking at the outcomes of their in vitro models.
Mm-hmm. Excellent, excellent. Well, thank you again. We really appreciate you joining us. We consider you a cytocentric visionary because of all your work to really put the cells and their well-being and their environment at the forefront of your work. And you have done amazing work now and found some really important findings, and that we're hoping that others will see how important it is to consider the actual cell environment as a critical component of their work as well. So
Great.
thank you again for joining us, and maybe we'll check in on you again in another year or so and see where you are then.
I would love to chat. That's fantastic. Thank you very much,
Okay.
Dr.
Thank you very much.